Hello, I want to welcome everybody to today's Gray Matter. I'm Kelsey Martin. I'm the Dean of the David Geffen School of Medicine at UCLA. It's wonderful to see we're almost at 100 attendees. I see a lot of names of people I know, of people I don't know, and here we are doing a virtual event. We're doing a virtual event because of the pandemic, and the pandemic has taught us something so incredibly important about what academic medicine can do in going from the most basic science to clinical care, where we have a hospital that's been taking care of COVID patients, setting up vaccinations, and researchers who've contributed to developing new therapies. We have decades of research here and across the country that have been critical to developing the vaccines that have made such a difference, and such a difference that probably, hopefully, the next Gray Matters will be able to be in person. So that power of academic medicine, I think there's nowhere that it's more evident than in neuroscience. Thinking about psychiatric diseases, brain disorders, we need a deeper understanding of the brain. We need to understand how the brain gets wired, how the brain functions. And we're so fortunate at UCLA that we have amazing individuals across our campus who have dedicated their careers to understanding at different levels of analysis the brain and how it comes together, how it works, what happens when it doesn't work. And you're in for a great treat today to hear from several of our faculty who are working in different aspects of brain development. So I want to thank all of you for joining. I hope that you really enjoy the presentations. I hope that you leave with a sense of excitement about the future of neuroscience and about the potential of research in neuroscience to really impact how we understand brain disorders. And with that, I'm going to turn it over to my colleague, Dr. Larry Zipursky, a faculty in the Department of Biological Chemistry and one of our true leaders on our campus in neuroscience, a Howard Hughes investigator, member of the National Academy of Science, the American Academy of Arts and Sciences, whose own lab has made amazing contributions to understanding how neurons wire together to create a brain. So with that, Larry, I'm going to pass it over to you.
Thank you, Kelsey. And welcome to this exciting program. I am delighted that you could join us for what I hope you will find an interesting conversation about the brain. The adult human brain contains 100 billion neurons. Neurons are cells, and these cells, in magical ways we are only now beginning to understand, allow us to perceive the world around us, make sense of it. They underlie our ability to think, to feel, and to communicate with one another. They form and hold our memories, and in a fundamental way, the brain is our identity. How the brain takes in the world around us, makes sense of it, and indeed changes it, is arguably the most fascinating scientific question of our time. The extraordinary capability of brains to accomplish all this lies in four fundamental features of neurons. First, there are tons of different types of neurons that are highly specialized to carry out specific tasks. They all have the same genes, but mysteriously use them in different ways. Second, these neurons are connected to one another, forming networks of cells that act together to create specific behaviors. Third, neurons receive and send information through a specialized electrical and molecular language. And finally, these connections are malleable. They are built to adapt to the contingencies in the world and are profoundly affected by our experiences, particularly our experiences during childhood and adolescence. We are products of both nature and nurture. The amazing capacities of the human brain are the product of hundreds of millions of years of evolution. The UCLA neuroscience research team is comprised of scientists and physicians in the School of Medicine, as well as faculty within the schools of engineering, life sciences, and physical sciences. The goal of the theme is to support cutting-edge research initiatives to understand brain function, development, and disease, and to convert this understanding into therapies. The Gray Matter series, of which this afternoon's event is a part, highlights different areas of brain sciences at UCLA, and today our discussion focuses on how brains develop. I'm particularly excited about our program for two reasons. First, my own scientific interests are focused on this problem. I've devoted my career to understanding how brains are put together. This is a fascinating scientific problem. It is also medically significant, as abnormalities in brain development underlie neuropsychiatric disorders such as schizophrenia and autism. The second reason for my excitement is that we have four exceptional scientists joining me today: Aparna Bhaduri, Josh Trachtenberg, Dan Geschwind, and Adriana Galván. Their research is absolutely fascinating and spans human brain development. Aparna studies the earliest stages of brain development through new and exciting breakthrough technologies. Josh has pioneered studies on how our brains are influenced by early life experience. Dan has revolutionized our understanding of autism, and Adriana has made key discoveries in how the adolescent brain develops. Each of our panelists has pre-recorded a short introduction to their work. This will then be followed by a Q&A session. Some of you have already submitted questions, and I have thrown in some of my own. Please also write in the questions in the Q&A function in Zoom, which is at the bottom of your screen. It's a pleasure to introduce our first speaker, Aparna Bhaduri. Aparna is an assistant professor of biological chemistry. She joined the UCLA faculty just last January. Already at this very early stage of her career, she has made seminal contributions to our understanding of the earliest stages of human brain development.
We have long been interested in studying how the human brain develops, but we cannot directly study the human brain or perform experiments with it. Luckily, there have recently been two exciting revolutions in the field. For the first time, we have the ability to use stem cells in a dish to make parts of the human brain. Stem cells are, in theory, capable of making any cell type in the human body, but only recently have scientists been able to harness these stem cells to grow in a dish. Within the last few years, scientists have discovered that in the right conditions, we can make what's called an organoid, or a stem cell-based model of an organ. In my case, I am fascinated by cortical organoids, or models of the human cortex. The human cortex gives us many capabilities of perception, judgment, and cognition, and contains many different cell types. The organoids are able to make many of these diverse cell types and allow us to study the fascinating questions of how initially uniform stem cells can give rise to so many unique cell types. This allows us to start to look at how human brain cells communicate with one another and how they can influence each other during development. With the organoid system, we can now start to ask questions about how normal development of the human cortex occurs, how it may go wrong in cases of neurodevelopmental disorders using cells that resemble actual patient cells, and how we can leverage the potential of these cells for the treatment of injury or neurodegenerative diseases. This is revolutionary because for the first time, we can perform experiments in human brain cells. One thing that is simultaneously mesmerizing and challenging about the human brain is that there are so many different types of cells. Each cell has the same DNA, but turns on different parts of the information within DNA to make it unique. Another technological revolution, single-cell sequencing, has allowed us to study all of these cells in a remarkably simple way, one at a time. The idea that we could study the differences between different types of cells in normal and diseased cortex was inconceivable even 5 years ago. We can now ask what genes any individual cell turns on or off. This allows us to carefully understand the rich tapestry of cells that exist both within the human cortex and the cortical organoids. and to understand how one cell type begets another, or how a particular disease uniquely influences one cell type but not the rest. Using these exciting new technologies, my lab is trying to understand how the normal human brain cortical development generates each part of the cortex with a unique function, and also how we can leverage organoid and single-cell technologies to develop tools to combat disease.
Our second speaker is Josh Trachtenberg. Josh is a professor of neurobiology. He developed ways to peer into the developing brain to see how neurons alter their shape and function in response to changes in experience. These studies have profoundly influenced how we think about the developing brain.
The goal of the research in my lab is to understand critical periods of early brain development. A baby's brain is not just a smaller version of the adult brain. Many of the brain regions that we most associate with being human, those that give us our perception of faces, those that underlie language, and those that give rise to our social intelligence, their development is exceedingly sensitive to the kinds of early experiences we encounter in childhood. Our early experiences actually change the wiring of the brain, and the brain is most susceptible to and instructed by these experiences from birth until about puberty. And these are known as critical periods of early brain development. And when critical periods end, the brain becomes far less affected by new experiences. Anyone who has been around kids knows how quickly they learn and with such apparent ease. A great example of this is language. There are about 6,500 languages in the world, and children learn languages simply by being exposed to them. There's no didactic lecture given to kids. The brain is intrinsically wired up to process sounds as words and to process a series of words as language with meaning. But the specific languages we are exposed to in childhood sculpt this neural circuitry of these language areas. And if you don't learn languages during this critical period, it's quite difficult to learn them thereafter. And this doesn't only apply to language areas, it applies to every region of your cortex. Our early experiences are fundamentally important. This interaction of this intrinsically established neural circuitry and our specific sensory and motor experiences of our youth drives that age-old question of nature versus nurture. That is, how much of who we are is determined by our genetics and how much of it is determined by our early experiences. This is a profoundly important question to understand. How much can we change about who we are? How important are these early childhood experiences? Can we overcome early traumatic events? And ultimately, why does the brain lose this remarkable plasticity? And can we turn it back on and ultimately rejuvenate the brain? In my lab, we address these questions using specialized microscopes and lasers to image thousands of individual brain cells in the living brain and to measure their activity and their connectivity across development. By doing this in mice, we can manipulate not only their experiences, but their genetics as well, and gain real insights into the interplay of nature and nurture as they sculpt and shape the function of and connectivity of developing neural circuits.
Our third speaker is Dan Geschwind. Dan is a professor of neurology and senior associate dean and associate vice chancellor of Precision Health. Dan's work has revolutionized our understanding of autism and other neuropsychiatric disorders by utilizing cutting-edge technology to understand the role of genes in these disorders.
It's great to be here with all of you this afternoon. I know this is going to sound odd, but I'd like to start my brief introduction to autism by talking about the tummy. Actually, ulcers. These are sores that are in the stomach or small intestine that can cause chronic pain, bleeding, and sometimes death. When I started medical school, we're taught that ulcers were due to lifestyle choices, spicy food, and the inability to handle emotional or physical stress. In fact, there were papers attributing ulcers to dominant and obsessive mothers. This was not so different from Bruno Bettelheim's 1966 framing of autism in his book, The Empty Fortress, where he blamed dominant and obsessive mothers for autism. And of course, when we get back to ulcers, the treatments included avoiding stress, blocking acid, a bland diet. But this didn't work. Symptoms would sometimes go away, but the ulcers always returned. This all changed when Dr. Robin Warren and Barry Marshall found a new bacteria that caused ulcers. It took almost 15 years for this to be accepted, but they got the Nobel Prize in 2005 for this. The lesson here is that finding the true cause totally changed management and changed outcomes. The same is likely true for autism and other disorders. When I started work in autism about 20 years ago, there was almost nothing known about the causes of autism. There were all kinds of theories, and sometimes these led to very dangerous interventions. I thought perhaps because we knew that autism had a strong genetic basis, that if we could identify one genetic cause, it would be a productive career. Fast forward now 20 years, now thanks to advances in basic science and genetics, we're on the cusp of a revolution in our understanding of the disorder. We have now identified more than 100 different genes that cause autism and related neurodevelopmental disorders. Genetics gives us a starting point for understanding disease mechanisms. Please don't get me wrong, genetics is not deterministic, but genetics gives us a toehold to beginning to understand mechanisms. The way that it works is that with genome engineering, we can actually put mutations or disease-associated variants in model systems, such as flies, worms, fish, or mice, even in a dish in human-derived stem cell mini brain cultures. We can put in the mutations because these systems are all easy to genetically engineer and explore behavior or biological changes that occur during development and during the function of that organism. We and others have done this, and we've begun to identify the mechanisms of disease in some specific cases, and this has led or will lead to specific clinical trials in the near future. Genetics has also shown us where and when most autism begins. The genes involved are expressed most highly during early brain development and in neurons that connect regions of the brain. This shows us what the brain basis of autism is. It's also shown us that autism is very heterogeneous. It's a collection of rare disorders, not unlike cancer. That provides challenges, but it also shows us that with this knowledge, we have serious hope for new therapies.
Our final speaker is Adriana Galván. Adriana is a professor of psychology and dean of undergraduate education. Adriana has made key discoveries about brain development in children and adolescents, including the importance of environmental factors in regions of the adolescent brain associated with risk and reward.
Thank you for the opportunity to talk about the developing adolescent brain. Adolescence in humans is the second decade of life. Typically begins at the start of puberty around ages 9, 10, 11, or 12, and goes through about the early to mid-20s. And so this is a significant time in life. There are a lot of changes. There are changes physically, biologically, emotionally, and certainly neurobiologically. And what we've learned in the past few decades is that the adolescent brain undergoes many more changes than we used to think. We used to know that the brain underwent a lot of changes right after birth. But now we know that there's a second wave of what's called plasticity in the brain that occurs during adolescence. And during this time, there's continued refinement of the prefrontal cortex, the brain, the part of the brain that's right above your eyes. There's continued refinement of what's called the mesolimbic circuitry. And this includes brain regions that are responsible for reward, for learning, for emotion processing. All the things we know that are characteristic of adolescence happen in this brain region. And most importantly, what happens during adolescence is that the connections between regions and among regions also changes. And what ends up occurring is that brain regions are better able to communicate with each other. This leads to enhanced efficiency for brain processing. This leads to increases in neuronal functioning. And most importantly, it helps adolescents respond to the changing world around them. You probably know, if you have an adolescent or know an adolescent, that there are a lot of psychosocial changes that happen. People are more likely to spend time with friends outside of the home, to explore their identity, to pursue new passions. And all of this is supported by these changes that I've described in the brain. And so while sometimes it could get adolescents in trouble, and adolescents get a bad rap for engaging in high rates of risk-taking, all of it contributes to the important need they have during this time to explore their world and to form their identity and to explore different identities. And we're very lucky as a human species to have a period in development when we are naturally inclined to learn, to establish new social bonds, and to explore the world. Thank you.
Thank you, and welcome Aparna, Dan, Josh, and Adriana. We have plenty of time for questions. Please, to the audience, please type in your questions in the Q&A function. In Zoom, it's on the bottom of your screen. As I'm sure most of you know, there are various viewing options in Zoom, and we suggest you use the gallery option for this Q&A session. So I'd like to start this off with a question for Aparna, and talk a little bit about this wonderful technology, organoid technology that you described, which really has revolutionized biology. In part because it provides an incredibly powerful and safe way to study neurons and groups of interacting neurons from patients. I was wondering whether you could tell us how you make organoids from the cells of virtually any patient you wish.
Yes, absolutely. There's a lot of questions, as Dan was mentioning, about how you can have different neurodevelopmental disorders emerge during time points of development before we even have a chance to look at an individual or look at their brain. And so what this technology allows us to do is actually take a sample of somebody's skin and take that, and because it has the exact same DNA information that the cells had when the brain was developing, to basically turn off the skin genes, turn on the stem cell genes, and then put them in a dish. What we do from that point is actually quite straightforward. When they're in a stem cell format, we're able to count out 10,000 cells, put them in a small plate. We usually have about 96 wells in that plate, and then we put them in liquid that gives them instructions. We put certain different compounds into that liquid that gives them those instructions to start taking on those programs of brain development. And that way we can instruct the cells to start making the cortex. This then allows us to look at what is similar or different between the individuals who suffer from neurodevelopmental disorders or from other individuals who maybe don't have those phenotypes.
Great, thanks. So Dan, just a sort of follow-up question from you, for you. But one of the real challenges in studying human brain diseases is getting access to the developing human brain and to carry out experiments on it. Can you tell us a little bit about how organoid technology that Aparna just described lets you study autism and other disorders, such as schizophrenia, in the lab?
Yeah, it's a great question, and I really, it's really exciting to have Aparna here joining us, because that we're beginning to build quite a large community in this area and have some really ability to do cutting edge studies together. So I'm very excited about that. You know, essentially, there are two major questions, and one is, how well do these developing brain organoids recapitulate in vivo brain development? And we and others have actually begun to ask those questions, and the answer is, up to a point, really pretty amazingly. They do many, if not most, of the things that are going on in vivo. One has to kind of put them together to be able to do that and engineer them to do that. It doesn't just necessarily happen, but and that's very exciting. So it's an amazing model. And since, as I mentioned, we now know that many autism risk genes act during early development at the time when neurons are being born, which is prenatal, we can model that in vitro by using genetic engineering technologies like CRISPR, which make it quite easy to put these mutations into these cells and grow them and see what their effects are and measure them using all of the techniques that Aparna just mentioned. And more, we can do physiologic measurements, molecular genetic and cellular measurements, and even as the technology is moving, even begin to look at connectivity between different kinds of cells. So it's very exciting. Now, for later onset brain diseases, there's still some more work and there's some frontiers that need to be broken. Although when we looked at when and where let's say, genes that predispose to schizophrenia are expressed, they are expressed in general a little bit later. There are some that are expressed in early brain development and some a little bit later, but many of them are expressed at high levels in these in vitro systems. And so I think these new brain in a dish systems make things very tractable. And we can use genomics and RNA sequencing and other methods to compare what's going on in a dish to what's going on in vivo with brain tissue, and actually do this in a very rigorous and quantitative manner now, which wasn't possible 10 years ago.
So Dan, thanks. I'm just looking through this fabulous set of questions we have in the Q&A, many of which will get us back to some fascinating questions about autism. I just want to briefly jump to critical periods to talk a little bit about that with Josh and Adriana before we come back to autism. And at least for me, critical periods, Josh, that you study are among the most fascinating phenomena in neuroscience. Especially, these are special periods during development where brains are particularly influenced by our experience. Why are critical periods so important in human brain development?
Well, in humans in particular, we have a pretty heavy lift once we're born. I mean, our brains are so large that we are forced to be born at a very, very early stage of development. And as a result, a large number of connections in our brain are still being formed. And these connections are shaped for many, many, many years after we're born through our experiences. And so what distinguishes the critical period from adult learning is that, as an adult, as a teenager in high school, or as a college kid, or as an adult, we know that almost everything we learn requires reinforcement. We have to get, either punished or rewarded for learning. And that's what drives learning, is we've got to pay attention to things and we have some sort of reinforcement. When babies are born, those mechanisms are sort of, not really well developed. They have to learn just the basics, how to, build an entire repertoire of objects that they can recognize. They recognize the basic properties of matter. You can see this in the classic cup trick, where young babies, if you put a ball into a cup, they think it's gone. And at some point, some months later, they realize that the ball is still there. So they've realized, without any instruction, that matter has certain properties, and they've learned things about, the properties of objects, or that certain sounds, certain phonemes are in fact words and can be used in complex ways. So they've had to build up this immense understanding of the world around them without any real instruction. It's a self-learning machine. And so how does this circuitry, how do you build a self-learning circuit? And then why does it change? Like, what is the advantage of changing the circuitry, one that requires reinforcement and attention? So you guys mentioned things about different cell types and the strengths of using these organoid systems to study all these different cell types. And yes, this is fundamentally important. Like, how do these cell types change as a function of development? How are they engaged by our experiences? And how does this engagement allow these brains to sort of self-teach? It's a fundamentally important question. We're still trying to get at that.
Yeah, I mean, I'm endlessly fascinated in critical periods, and I have to say, the one that I find particularly fascinating and not clear how one studies it is really that critical period in human development during the teen years. And we all remember that as teens, and also for those of us who have had kids who've gone through teens or in teens or about to enter teens, and have thought about this part of human development. And I wonder, Adriana, if you could tell us a little bit about sort of what are the key neurobiological changes? What's happening in a teen's brain?
Yeah, it's such a good question. And in the past few decades, we've gotten some insight using brain imaging technology whereby we ask adolescents to come to the lab and we scan their brain while they're doing different tasks. And Josh really set it up nicely to say that when you're born, you are, in the words of Esther Thalin, a great developmental psychologist, you're building something out of nothing, right? How does that happen? How does the child construct their brain based on inputs? And in adolescence, of course, the brain is more built up. They know how to do the basic things like walk and talk and bond. But in adolescence, we're refining those connections that started to get set up in the initial critical period. And we know that certain brain regions are pretty well developed by adolescence. So the brain regions that control motor behavior, for example, are pretty well done, maturing or developing. But the brain regions that continue to grow are those that are involved in executive functioning, in reasoning, in planning for the future, and also those brain regions that support learning. And that reinforcement that Josh mentioned of learning from reinforcement or from punishment, these systems continue to change during adolescence in ways that sometimes lead them to have higher reward sensitivity or higher reward learning than people of other ages. But they're critically important to helping them launch and transition into adulthood.
Thanks, Adriana. There are a bunch of really cool questions here that actually raise some interesting questions about mechanisms, what could be happening in the teen brain or the early child's brain. I wanted to read one of these for you, and I'll leave it to any of you to pick up on it, or perhaps a couple of you want to comment on it. So the question is the phenomenon of synaptic pruning. So that's like removing synapses, connections between cells, at certain times in brain development, for instance, early childhood, early adulthood, has been proposed as a natural event that has gone out of control in individuals with autism, onset of 2 to 3 years, and schizophrenia, 18 to 22 years. Is there any research that has confirmed this hypothesis? And if so, are there any developments or treatments for these conditions? So I think this is this fabulous question. I'd love any of our panelists to pick up on it, whoever would like to comment on it, and I'm sure a couple of you would.
This has a rich history, and the phenomenon of synaptic pruning is found basically anywhere you look in the brain, including the peripheral nervous system. One of the greatest examples of this is at the humble neuromuscular junction, where in adults, each muscle fiber is innervated by a single motor neuron, but in very young animals, each muscle fiber is innervated by up to 10, 20 something numbers of motor neurons, and there's some sort of competition between them. People then looked at this in the central nervous system, and they found if they counted synapses, that synaptic number was dropping, in fact, in adolescence. And this gave rise to the view that our experiences are sculpting the brain. That is, we have we sort of massively overconnect things, and by our day-to-day experiences and in the world, and by using these circuits, we can eliminate connections that don't quite work very well and strengthen connections that do. And by doing this in the sculpting process, you start with this big piece of marble, and by diligently cutting away at it, you reveal the pietà, if you will, right? These models get refined with time. I'm hoping to refine some of these models, but they're still there. Look, synapse numbers are changing. It's not quite, in my view, so cut and dried that experiences are just sculpting connectivity in the brain. I think there's a great deal of interaction between genetics and experience that determines, what's happening to these synapses, which ones are strengthened. But in terms of how these affect disease, Dan, I'm going to defer to you on that.
Well, I could give a short answer, which is it's actually not known, but we're getting close to knowing it, because we can use all of this basic neurobiology that's being studied, all the way from the microscopic, like the work that you do in synapses and the work that people do in developing and setting up of synapses, all the way through to macro or mesoscale, like imaging, that gives us functional pictures about how things are going together and working. And we can look at autism or disorders like it in that context. And it's fairly clear that some of the mechanisms that are involved in sculpting synapses and that are involved in neuroplasticity are awry in autism brain. And we've shown that. We've actually defined what I call the neuropathology of autism by looking at postmortem brains and seeing a massive upregulation in a bunch of different kinds of glial cells that reduce kind of certain synaptic molecules, and a reduction in some of those molecules in the brain. Now what we have to do is kind of connect that all the way back to the cause. How did that come about? Is that adaptive? Is it maladaptive? But the point is that these fundamental basic discoveries about neural development and about neuroplasticity are essential to putting what we're finding in autism in context. So we're beginning to define what's going on in the brain, define mutations, but the only way to understand what they're doing is via exploring what they're doing at the basic mechanistic level. I hope that makes sense. Larry, did that make sense?
Yes, it definitely makes sense. And I was, as you were talking, I was looking through the Q&As, and there was one that I thought was sort of an interesting question I hadn't thought about before, but is really related to what you and Josh talked about. And I think Adriana perhaps has something to say about this. And that is, we talk about critical periods, we talk about sculpting, we talk about pruning. And the question here is, what is the difference in the plasticity of very early life and that of an adolescent? Are they really the same sort of process, or is there something fundamentally different? I mean, one thing that's always intrigued me is there's sort of a clock for this adolescent stuff, right? And whereas the early development seems to be a continuum from birth. Anyway, I know nothing about this area.
Yeah, it's such an interesting question, and one that scholars in the field really focus on. And one thing that is certainly different is that the context under which the plasticity is happening is very different. So when you are starting from a very new brain early in life, there isn't a developmental history to contend with. There isn't already learned skill typically that is working with the new plasticity. And so that's one thing that is different. The other thing is that there are already different maturational processes happening in certain brain regions as an adolescent. And so although some of the basic mechanisms may be the same about, at the neuronal level, how plasticity happens, how we acquire new skills, the context is different. But I'm curious to hear what Josh thinks about that.
I think it would be great if we could bring Aparna in on this discussion, actually.
So yeah, I was thinking the same thing because I think, one of the problems we have with humans, we're endlessly fascinated with the human brain and how it develops. Mm-hmm. But we can't do experiments. We've not really been able to do experiments to ask, well, what happens if you screwed up with wiring in this way, like we can with experimental animals? And up until very recently, it's all been doing your experiments in a mouse or a fruit fly or a worm, C. elegans, and then looking at whether some of the same genes are also found in humans. And I think maybe Aparna could talk a little bit about the promise of what these organoids can do to actually directly address questions of developing brain in human.
Yeah, absolutely. And I think, it's a really interesting discussion. How do these different maturation events sculpt the different connections between parts of the brain that ultimately lead to what is eventually the adult brain? But a lot of things happen between early development and then. And one thing that's really exciting for us is that for so long, the human aspect of studying brains has happened at an observational level. So people have drawn pictures of it, they've dissected it, they've looked at it, but they haven't been able to actually change things in it. And so I think what's really exciting, and even though the organoids are aimed to model early stages of development, myself, Dan, a lot of other people in the field are really doing whatever we can to accelerate that development, to be able to study early childhood periods, even hopefully one day some of the adolescent periods, to study how do different genes, the ones that are genetically identified, have changes in specific cell types. And so one way that you can do this is you can change the genes in a particular cell, and then you can try and connect it to other parts of the brain. So I talked a little bit about cortical organoids, but you can make organoids for different parts of the brain, and those brain regions communicate with one another. And as we're hearing from the others, that communication is really important for this learning and that maturation of those circuits to develop those skills of judgment and motion and all of the things that we need to be doing as individuals. And so that's one direction where this is becoming very exciting, and we can look at the human specifically.
Yeah, if I could add to that, is that all right?
Yeah, definitely, please.
Yeah, no, I love that. And one of the things that, you know, that the field has recently identified is that you can grow these organoids in a dish, and they have an intrinsic clock that's kind of, it seems to be, that's mirroring normal human development, which is really great for modeling more accurately, human neurodevelopmental disorders that are early. But it also poses a challenge in that it takes 9 months. But after about 7 or 8 months, we see that these organoids at a genetic level, at a molecular level, and even at a protein level begin to resemble postnatal tissue, which is great. But you know, it takes 9 or 10 months to get there. And if you want to model, let's say, Alzheimer's disease and other things, you're going to have to make other modifications. So one of the challenges in the field that we're working on at UCLA and Aparna and other, it takes a village, is to make these, make development happen more quickly, and aging as well, so we can study adolescent brain in a way. The adolescent brain is so mysterious. It's hard to imagine we'll be able to study that in a dish.
Yeah, sort of modeling risk behavior in a dish. I think that's a wonderful challenge. So, all this discussion about plasticity and sculpting circuitry and behavior reminded me of this. Actually, the second question we had on our list today from someone from the audience, and I'm sure for many people this question is an important one. So, what connections? Let me see where this is. Hang on, here we go. How flexible is neuroplasticity? Can an adult really learn completely new skills, or at least deeply modify ingrained behaviors? And I was sort of thinking about this as Josh talked about language and how easily children learn languages, and how painful it is for those of us who are long past that critical period to learn, frankly, anything, but certainly a new language. And what do you think the prospects are of really learning lessons about plasticity during early development, and whether those lessons will be applicable for older individuals to enhance, or I should say rescue plasticity?
Well, it's a double-edged sword, that question, right? So turning on the plasticity that characterizes, infants to, toddlers to very young kids, that kind of plasticity is set up to really extract all of the rules of the world around you and build a working model of the world around you that you can then act upon, and to gain things like abstraction. The problem with it, as we know from critical periods, is that it can also rapidly degrade, right? So if during critical periods you aren't exposed to these things, that circuitry doesn't work, right? It doesn't get built. Secondly, if you start building that circuitry and you successfully build that circuitry and you're still in these critical periods, you can then rapidly degrade that circuitry and go right back to where you started, sort of not quite the tabula rasa, but you would have this degraded circuitry. So I don't think it would be advantageous for us to recreate that environment in the adult brain. There are, of course, a number of ways that people are exploring to enhance learning in the adult brain. I hate to sound too Southern California, but probably exercise is really the best way to promote learning in the adult brain. Exercise more and make sure your diet is quite good. Again, I'm not even from Southern California, but maybe I've lived here too long. Those two things are some of the best things you can do to really enhance brain plasticity. Exercise is tremendously important, and just try new things and keep doing new things. Travel, get involved in different things, get out and about. I know it sounds contrived to say that, but that's what
It's true, but it's true. So it's okay for you to say that. And get good sleep. That's the other one, right?
Absolutely.
Mm-hmm.
So we have let me just look at this Q&A. We have quite a number of questions, some of which touch on COVID, and it's sort of inconceivable that at this time in history we shouldn't talk a little bit about COVID and the brain. And I wanted to ask Aparna whether there's any evidence that COVID infection affects the developing brain in utero, and if so, would organoid technology provide a way to study this problem?
Yes, absolutely. This has been an important concern for many expectant mothers during the COVID pandemic, and it hasn't been long enough that we actually know what the impact might be on someone who has a baby during this time and contracts COVID. So there are a number of researchers. I personally haven't been involved in this work, but there has been some exciting work done in the field looking at COVID infection in cortical organoids. And what people have observed from this is that there are some cell types that seem to be affected by COVID virus. And this is interesting, and I think is important to know that if the virus were to transmit from mother to child, that it could have an impact on brain development. These aren't conclusive answers, but this is an opportunity to better understand how it could be happening, and also help people who are in this situation make some decisions about whether or not they would take some risks or not, use more safety measures, and also whether or not to take the vaccine, which currently public health officials are recommending for pregnant women. And so these are important questions to be asked. And I think there's a precedent for this as well. There was a Zika virus pandemic a couple years ago, and although very fortunately that one flamed out pretty quickly, that one had a big impact on developing children, and organoids were one way to study exactly how that was happening, and also to start to develop treatments for it, which luckily weren't fully finished in their clinical trials because the virus largely flamed out.
So Dan, the flip side of infection is vaccination, and I wondered whether you could address this issue in terms of vaccination for COVID, but vaccination also more generally for developing fetus.
Yeah, number one, get vaccinated. No, I want to follow up on a couple things that Aparna said, and then I'll slip into this. With regard to Zika, it's actually, there's a UCLA study of mother and infant dyads published in New England Journal of Medicine that really outlined a huge amount of, and actually taking place with children in Brazil by a Dr. Nielsen in pediatrics. And her group has also assembled a cohort like this for COVID. So I think these studies, as Aparna mentioned, are going to be extremely important. One difference, and again, we need to study patients, but we can study these in organoids as people have. And actually the Zika studies in organoids have been quite remarkable so far. But Zika's quite different in that there's not a lot of evidence in most cases for COVID actually transmitting transplacentally. So what happens is, but that doesn't mean there can't be a problem, because the infection causes maternal immune activation, which in and of itself could actually have an impact on the fetus, even if the virus doesn't infect the fetus or the embryo. So there's a lot to be done there. But I think, the way to suggest that it's much safer to get vaccinated than to potentially go through a severe COVID episode with severe cytokine storm and other things like that, which are likely have a high likelihood of potentially impacting the fetus. So now in terms of, it gets to a broader question, right? And we're talking about autism and neurodevelopmental disorders. I do want to make it clear that there's no epidemiologic or other strong connection after now more than 24 studies all around the world, multiple different study designs, multiple different populations that show any serious connection between vaccines and autism. That doesn't mean in a 1 in 10 million rare case where somebody has a mitochondrial disorder and then gets a fever because of the vaccine, that wasn't a causal thing. In fact, we know that can occur, but those are extraordinarily rare cases. And vaccination is not to be avoided because of concerns about developmental disorders, for sure.
Thanks, Dan. I wanted to ask Adriana maybe to comment a little bit on COVID and its effects on teens, and in particular, sort of the whole lack of one being stuck at home with their parents, not being able to attend school, not to be out with friends and exploring the world. Is this an area that's now being studied?
There are so many studies. A lot of psychologists and neuroscientists who study adolescents mobilized to launch nationwide and international studies on the effects of COVID on adolescents. And the jury's still out. Of course, we're still in the pandemic, and many adolescents are just now going back to school. But certainly, what you were getting at is that because adolescence is such a rich time for social interactions and social bonding, that certainly has been felt by adolescents and is missing. But what's interesting is that for adolescents who struggled with anxiety before the pandemic, some of the data suggests that they actually find they report lessened anxiety, and in part because they are now home, of course, and the things that made them anxious, like going to school and social interactions and things like that, have been avoided for almost a year. So it's really interesting. Another thing that has been, I would say, beneficial, but has changed during the pandemic is adolescent sleep. Adolescents notoriously do not get enough sleep as a cohort, and during the pandemic, instead of getting out of bed, an hour and a half before they have to be in school, they've been able to do that with less lead time, and so it's led to more sleep. So anyway, we don't know the effects, but certainly it's being studied.
Okay, well, I think, let me see if we have time for one. Looks like we have time for one more question. See if I can find one. Dan, in your presentation, you made a comment about genes being genetics not being deterministic. And maybe you could take a minute just to clarify that and what the relationship between genes and environment.
Yeah, so a couple things. One is, I think it's brilliantly exemplified by the kind of work that Josh and Adriana are talking about, that the brain has a lot of plasticity. In fact, that's how our behavioral and cognitive therapies that we employ in developing children with autism actually work. They're actually modifying brain function, and that can happen even if but to get back to what our brains develop along a track, and the genes that, let's say, can cause or increase risk for autism are genes that disrupt brain development. They're not kind of autism genes per se. They can lead to a number of different symptoms, intellectual disability or seizures or autism. They increase risk. So we think of genetics as being something that increases risk. Now, sometimes people have a Down syndrome mutation or other very large effect size mutations that increase risk dramatically. So we know that there's going to be a developmental disorder, but still exactly what that's going to look like and how we could modify that, that's where I think there's an enormous amount of promise, that once we understand what the mechanism of the genetic developmental impact is, the thought is we can intervene to modify that, because the brain has a certain degree of plasticity. I'm not saying that's an easy thing to do, and nor am I saying do we know how to do it now? But I think once we have a toehold with genes and mechanisms, we can begin to think about that. And and for most people, for most neuropsychiatric disorders, it's not a black or white kind of thing. They're somewhere on a spectrum of risk. It's called polygenic risk. It's a combination of the common genetic variation that we all share that just adds up, and with environmental factors leads to that condition in that particular person. And so I think we have to be careful when we're talking about genetics, and I don't want people to feel that it's once you have something genetic, that's it. No, that gives you a very, very important clue, and our hope is that once we have that information, we can modify development, modify the brain to to optimize patients' lives. I'm hoping that was clear. It's a very complicated question.
A complicated question, and probably not the best question for me to have asked at the very end. It's a really important question, and unfortunately, we have come to the end of our time this afternoon. And I'd like to thank our Dean, Kelsey Martin, for her support of the neuroscience theme, and particularly to thank Aparna, Josh, Dan, and Adriana for sharing your fascinating work and your thoughts on brain development with us. Thank you very much.
Thank you. Great pleasure.
My pleasure.
Thanks, Larry. Thanks, Kelsey.
And I'd like to thank all of you for joining us today. Many of you have been generous supporters of neuroscience research at UCLA. I was thinking of you yesterday as I read an interview with Dr. Anthony Fauci. He was asked if so much money is provided by the NIH for biomedical research, how important is private philanthropy? His answer was simple and unequivocal. The NIH does not fund high risk, high reward science. And it was precisely this type of science that led to the unprecedented speed, the miraculous development, I would say, of COVID-19 vaccine. Private philanthropy is key to the research ecosystem. And we in the neuroscience theme greatly appreciate your support. It makes a difference. If you're interested in more information about neuroscience at UCLA or philanthropic opportunities, please visit our website or contact Doran Esfahani. Thank you, and we look forward to seeing you in person at Gray Matters events in the not too distant future. Have a good evening.